Heterothermy, those fish that have much higher body temperatures than the surrounding environment, and we talked about how that happens. And so I just wanted to kind of hit on some benefits of that, and just to make sure everybody understands that. I know you guys saw these, and so I'm just gonna go through them quickly, but some of the things that happen when fish have higher body temperatures include the expansion of a thermal niche. So what that means is that they can occupy places that they wouldn't normally be able to do. And so because the environmental temperature is so cold, they're not going to be able to maintain a metabolism. And so it allows them to move into places where there could be a lot of food resources because they can maintain a body temperature that's higher than the surrounding water column. The other important part of this is that in animals that live in a variety of environments in terms of temperature, it allows their enzymes, we talked about the proteins and that, that many of which are enzymes, stay at a constant temperature basically. And so most of them, of course, most organisms have proteins that function optimally under certain conditions. One of those conditions would be temperature. And so if you can maintain those steadily, then those particular enzymes will work optimally. And so of course some of the tissues are more important than others, mostly around the neural processing part of it. The other thing that allows them to do is enhance, or it allows them to detect thermal gradients, right? So if you think about it, the only reason why we know the water or air is cold is because, or hot, is because we can contrast, right? So if the air temperature is warm and you jump into water that's cool, you can tell that that's cold because there's a difference between the air temperature and water temperature. So, but if you think about that in respect to what fishes are doing, if their body temperatures are always matching the environment, then they don't really have a good way of determining differences in temperature, right? So we can tell cold water because it's much cooler than 98. But if your body temperature is the same as the environment, then it's going to be more difficult to distinguish different temperatures. If your body temperature is elevated, then when you swim into colder water, you can tell that, right? And so that is going to be important, mostly because it allows them to find food resources and things like that, because these are ocean animals, and oceans have lots of currents that bring nutrient-rich water to different places, which enhances the zooplankton populations and things like that. So these animals are using those cues to find food, basically. And so if your body temperature is much different than the environment, then you're better able to distinguish differences in environmental temperature. Let's see. The other thing, of course, is that it increases their metabolism, and so for some of these organisms that have to have extreme bursts of energy to find or to catch prey, they might have to function anaerobically for a few seconds, and that ability to have a higher metabolism is going to be able to replace some of the molecular components to generating ATP. Of course, that come with increased respiration. So higher metabolic rates is going to enhance the ability to recover after you've functioned anaerobically for a certain period of time. So if you're an athlete, the best way to recover from an anaerobic experience would be to do what?
Ice bath.
What's that?
Ice bath.
Well, more acutely. Like when you run sprints, the best way to recover from a sprint is to do what?
Stop running sprints. Stop running sprints.
A, stop, and B, what?
Deep breath.
Rest.
Huh?
Rest.
Or rest, or
Eat.
I mean, like immediately after. Like you run a 40 meter dash time trial. The best way to recover from that is to A, stop doing it, and B,
Deep breath.
yeah, breathe hard, right? So what you're doing is but you can't do that unless your metabolism is high, right? If your body temperature is cold, you're gonna be going like this. And you will recover, but it will take 20 minutes to do it, right? So if your metabolism is higher, then you can do it much quicker. And that makes you, now you're available to do other stuff.
If you don't faint.
Okay, so if you have a higher metabolism, you can recover more quickly. Another thing, of course, that comes with higher metabolism is increased growth. Why is that better? You gotta think about the environment that these guys are living in. Why is getting big, fast, good?
Predators.
Yeah, because they're in the pool with a bunch of other predators that are limited in what they can eat by what? What are they limited by? Well, who's the main predator of a little fish?
Big fish.
Big fish. And so the big fish, most big fish are limited in what they can eat by what? What they can fit in their mouth. So gate limited predators. And so if you can only open your mouth this big and now the baby is this big, you can't eat that. So the faster you get to that point, the better off you are. You're gonna exclude a bunch of size classes of predators. We talked a little bit about this, increased neural processing. So again, these are high-speed swimmers and they have to respond quickly, right? So they're chasing down prey in schools and they have to be able to respond to that very quickly. And that means you have to see fast and you have to react fast. And that, of course, is gonna be impaired if the body temperature is low and you have slow metabolism. And so a higher body temperature is gonna increase that. And then, again, coming with the metabolism increase is an ability to digest food, store food resources much more quickly than you can otherwise. And so there's a lot of things that happen when an organism that is typically an ectothermic animal can generate body temperatures that are different than the surrounding environment, and in this case higher. Okay, so the next part of the discussion was coping with extreme temperatures, and so we talked a little bit about what happens. So at high temperatures, a lot of times the proteins get denatured. At lower temperatures, it could be a problem, not extreme low temperatures, but generally low temperatures, metabolism slows down. And so, of course, most fishes are adapted to periodic slowdown in metabolism, and that's mostly during the wintertime, depending on what kind of organism we're talking about. So different environmental conditions have different effects in terms of temperature for these animals, and fishes are better at coping with some of them than others. One of the things that's typical if you live in an aquatic environment is that the extremes of these aren't as prevalent as they are in a terrestrial environment. We touched on that a little bit. Water has a very high specific heat, and so it takes a lot of energy to change the temperature of it. And so air temperature goes way up, way down, way up, way down. Water temperature goes up a little bit, down a little bit, up a little, and slowly, right? So it's a little bit different if you live in water, but generally these animals are still exposed to variable air temperatures or water temperatures. Okay, so there's two scenarios we can consider in terms of coping with extreme temperature. One is extremely high, and then the other is extremely low. Extremely high temperatures, not generally a problem for fishes in terms of this. And that's because of other limitations for animals that live in water, right? So most fishes don't have any real adaptations to extremely high temperatures. And the reason why that's true is because when water temperature gets a little bit high, then there's some other physical consequences of that don't relate to physiology of the fish. And the main thing is oxygen availability becomes super low. Okay, so as a water temperature, before the high temperature affects the physiology of the fish, they run out of oxygen, right? And so essentially what's gonna happen at moderately warm temperatures, all the oxygen is driven off from the water column and the fish dies anyway. So most fish don't have any real specific adaptations to extremely high temperatures because they never experience that, right? They're already long dead by the time the water temperature gets to a certain extremely high. Okay, and so, and of course, other things drive that, the increased metabolic demand, because their metabolism is also higher. So all that is driving the oxygen or using up the oxygen. So they're never really exposed to really high environmental temperatures. On the other hand, there's some possibilities where you can have low temperatures. Of course, you know, most places on Earth have distinct winters, seasonal variation. So there's some things that can happen for fishes that are related to extremely low temperatures. And so when we're talking about extremely low, mostly what we're talking about is below zero Celsius. And so the main problem associated with below 0 Celsius is what? What's magical about 0 degrees Celsius? It's freezing. That's when water turns into ice. When liquid water turns into solid water. And so for endothermic organisms, not a problem, right? We are going to maintain a body temperature well above 0 degrees Celsius. But if you're an animal that's an ectotherm and you're living in the environment that is below 0 degrees Celsius, then there could be a problem, because animals, all living organisms are mostly made up of water. If water is freezing, then there's trouble. Okay, and so there's a couple of things that can happen. The intracellular formation of ice crystals, which is ice forming inside the cell. And that's a problem, of course, because it's going to damage very delicate membranes, organelles, and that's where all the machinery is going on. And so if you have organelles that are damaged, then they can't carry out their function, and that's gonna kill an organism. The other thing that happens when water turns to ice is that the amount of water decreases, which has an influence on the concentration of other stuff, right? What happens when when the amount of water decreases, the other stuff what? Increases. And so now your osmotic balance is off, right? So we are not only adapted to specific temperatures, we're also adapted, our enzymes are adapted to specific ratios of water to other stuff. If you take out some of the water by freezing it, then what's left is gonna be much more concentrated with the other stuff, and that's gonna disrupt physiological pathways that we have to maintain. And that's again part of what we would call homeostasis. If things are doing that, then it's hard to manage what's happening inside the cell. The other thing that can form, or that can happen, is you can have extracellular ice formation, which is formation of ice on the outside of the cell, which is not as big of a problem, because that's, you don't have nucleus and mitochondria and Golgi apparatus. You don't have that on the outside of the cell. That's all on the inside. It's still a problem, because it still changes the osmotic relationship between water and other things, right? So if ice forms in the plasma, then the concentration of water in the plasma now goes down. And if that's happening and the cells are bathed with plasma, then the water that's in the cytoplasm leaks out into the plasma. So now you have, again, the same problem. You've got a higher concentration of solutes inside the cell than you would otherwise, and that disrupts all the metabolic pathways. So there's really a kind of a limited set of circumstances where this is a problem. And that's mostly in environments that are not freshwater environments. So freshwater fishes are somewhat protected against the freezing of the plasma, and that's because of some physical components of water that's freshwater. And that is what happens to it when it gets to that temperature, right? So when water gets to 0 degrees Celsius, it turns to ice, right? That's pretty much well established. You guys probably knew that. But what happens to water when it turns to ice is it becomes less dense. And when that happens, then the ice crystals that form in fresh water do what? Huh? They float, right? So what happens is when freshwater environments that are exposed to extremely low temperatures, the surface turns to 0. And once that happens, you've got less dense ice forming on the surface, and all that forms on top. The maximum density for water is 4 degrees Celsius. And 4 degrees Celsius, because it's a maximum density, where's that gonna be? Below the ice on the bottom, right? So this maximum density pool of water is on the bottom. 4 degrees Celsius is 4 degrees above 0 degrees Celsius, right? So it's not turning to ice. There's no ice forming in the plasma of the fishes, because they're in an environment that's well above that. And so you've got this protective layer of ice that forms on the top. You've got, negative 10 degrees air temperature, but you have this kind of protective layer of ice, and it will go deeper the colder it gets, the longer it gets. But if you have a water column that's 6 feet deep or 10 feet deep, the bottom is always going to still be liquid. And these animals that are adapted to low metabolism during the winter are swimming around in water that's 4 degrees Celsius, because that's as dense as it's gonna get, that's always gonna be on the bottom. If it's only this deep, then what happens? It freezes all the way to the bottom, everything in the pool is dead, right? So they're not gonna be able to survive that anyway. So we're talking about permanent bodies of water that are relatively deep, always are gonna have some liquid water at the bottom. And so that, of course, is important because what's happening in the plasma of the fishes prevents ice from forming, basically. Okay, so essentially what's happening in the fish is that you've got other things other than water, right? So you've got pure water. If you just have H2O, it's gonna freeze at 0 degrees Celsius. But fishes aren't just water. They're mostly water, but not just water. Neither are any other living organism. So the solutes in the blood, everything else other than water, gives the freezing point gets it to about negative 0.7 degrees Celsius. So it has to be actually below 0 for that to freeze, because they're swimming around in water that's 4 degrees Celsius, or maybe it's 3, or maybe it's 2, or maybe it's 1. It's not negative 0.7, right? So they will never experience a situation where the water in their blood will freeze because of that. And so these guys don't really have to worry too much about their blood becoming less than that and causing ice to form. But those aren't the only conditions that this becomes important, right? So for marine fish, it's different, right? So for marine fish, especially ones at high latitudes, they're exposed to water temperature that is below their body temperature, right? Below 0, below negative 0.7. And that's because of the high concentration of solutes in the surrounding water column. Okay, so because there's so much salt in some of these places, not everywhere, but in some places there's so much salt that the freezing point is negative 1.86. Okay, why is that a problem?
Still below the freezing point of fish.
It's below the freezing point of fish. So there's liquid water that is negative 1.87 degrees Celsius. And this fish is now free to swim into it, right? So, which is different than freshwater. Freshwater freezes at 0. There's no liquid water that's below that, right? So the fish can't swim into a water temperature that's below 0. In the marine environment, they can, and because that is still liquid just above that temperature, but the freezing point of fish is still negative 0.7 degrees Celsius. And so that's well below the freezing point of the fluid that's in the fish. And so that's a problem, right? Because as these animals are in that environment, they're exposed to conditions that are cold enough to cause ice to form. And so all of them have some type of mechanism that will help them avoid that. One of the things that many of these fish will avoid is coming in contact with ice. And the reason why that's true is because ice typically needs some type of, especially in living organisms, needs some type of kernel to grow on. And so if they're never touching any ice crystals, like physical ice crystals, then it's harder for ice to develop in the cytoplasm of the cell. And so that's one of the behavioral things that they do. But that's not always the case, right? So there are some fishes that specifically make their refugia in and on ice. This is a goby, and gobies are famous for living in crevices and burrows and stuff like that. These guys are Arctic species, and they form their burrows in solid ice, and so they're obviously in contact with it. So you have to have some other thing. The main thing that these animals do is they produce specific types of molecules that essentially lower the freezing point of the plasma. And those are called cryoprotectants or antifreeze type material. And so typically they're mostly made up of proteins or glycoproteins. So proteins are amino acids, and then a lot of them have sugar components to it, and that lowers the freezing point. It also protects the cells from damage of physical ice forming. And so one thing that happens is if you get some ice crystals forming in the plasma particularly, these things will cover those structures, and so that they're not as big of a problem. It also, they also increase the concentration of, they produce solutes, right? So what's happening is they're trying to balance the movement of water. So if these solutes are now in the plasma, then water is gonna move across the membrane less freely than it would as ice crystals form in the plasma and solutes go up, right? So they're matching the increase in solutes that happens when ice forms in the plasma. And that reduces the movement of water across the membrane, and that helps to maintain some of the physiological processes. It also, of course, lowers the freezing point, and so that ultimately what's happening is ice is not formed in the plasma. Okay, so relatively limited circumstances, right? So most freshwater fishes don't have to deal with this because all freshwater fishes, because they're exposed to water that's always going to be above the freezing point. But in some conditions that would not be the case, and so these animals have specific adaptations to help do that. Okay, the next part of it was osmoregulation and excretion, and so the main part here is that these animals are trying to maintain the relationship between water and solutes in a constant manner, and there's different ways to do that. And so obviously it's going to be very important. And so a couple of terms that you need to be familiar with. Most fish are osmoregulators, which means they are actively regulating the amount of water in relationship to solutes. Okay, so on this particular diagram here, these graphs represent two different types of organisms. One of them is an osmoregulator. Or one of these lines represents an osmoregulator. Which one is it? Is it the horizontal one or the diagonal one?
Horizontal.
It's the horizontal one, right? So this is the environmental concentration of solutes. This is the concentration of solutes in the blood. So if you look at this animal, no matter what the concentration is, the concentration in the blood is the same. On this one, it changes exactly the same way as the environmental temperature. Right? So this is an osmoregulator. This is an osmoconformer. So these animals are just conforming to the environmental concentration of water and solutes. And that's what this is, right? So this is an osmoconformer, this is an osmoconformer. This is an osmoregulator. There's different ways that can happen. There's different tolerances for different types of fishes. And so some fish can tolerate only small changes in salt concentration. And so haline refers to salts. Steno means narrow. Uri means broad, right? So a stenohaline animal is an animal that has a very narrow range of salinities within which it can survive. And then a euryhaline animal is one that can tolerate large changes in salt content, right? So I don't know what's coming next. And so this kind of represents some combinations of things. And so I just kind of want to make sure we're familiar with these terms. So an example of a euryhaline fish would be something like, well, just about every fish that lives in estuaries. So like a sheepshead minnow or a long-nosed killifish. Those animals are euryhaline. They live in estuaries, which is the place where you'd expect salt concentration to change quite a bit, right? So if you get a lot of rain in the estuaries, there's a bunch of river water dumping into the marine environment, salinity goes down, they're fine. If you get no rain, lots of evaporation, salinity goes way up, they're fine. They can regulate their body temperature, or their osmoregulation in a precise way, no matter what the environmental salt concentration is. An example of a stenohaline organism would be something like a bluefin tuna that's always in open ocean water, 32 parts per thousand salt content. You move it into fresh water, they're done. Just because they're very strictly maintaining relationship between water and salt content. They can't tolerate much change. Okay, so this particular graph shows some kind of combinations that I want to make sure everybody understands. Right, so this guy right here, what are we going to call this guy? Is he an osmoregulator or osmoconformer?
Conformer.
He's an osmoconformer. Is he stenohaline or urihaline? Uri, right? So he can tolerate, they're living in a relatively broad range, but they are changing with the change, but they can do that over a wide range. This guy right here is also an osmoconformer, but he's what? He's stenohaline, only over a very short range. And then these guys are osmoconformers. This is a urihaline osmoconformer, and this is a stenohaline osmoconformer. And so you can expect to see some graphs like that on not this coming exam, but the next one. Okay, so for fishes, the most important organ really is the gills, and what's the main reason why that's true? Why would you expect gills to be so important in osmoregulation?
Ammonia.
What's that?
Ammonia comes off.
Ammonia comes off, but why is it the gills that are so good at that?
They're the ones that contact with water,
They're the ones that they're in contact with water, and there's a lot of
surface area.
right? So a huge portion of the bloodstream is always going to be directly exposed to the environment at the gills. That's why it's so important for them, and they can use it fairly easily because they're constantly exchanging with the environment. For other organisms like us, we only have a limited option in terms of doing that. We can't excrete ammonia directly into the air. It's not soluble in air, and so we have to do some other stuff. These guys, because they live in water, can use the gills easily to get rid of some things that will influence what's going on. Right, so typically, one of the main things, of course, is ammonia, and that comes as we break down proteins, so we have to get rid of it, and typically that's going to happen at the gill tissue because you don't have to, it's toxic, right, but if you can get rid of it quickly enough, then you don't have to do anything differently to it, and so you don't have to convert it to something else. So obviously that's going to require a mechanism for that. So if you're in water and most of the bloodstream is exposed to water over a really short time period, then you can use the gills. Okay, the kidneys also play a role, of course. What role they play is depending on the organisms that we're talking about. And we talked about this a little bit already. The kidneys, of course, the functional unit of that is the nephron. And so the blood is delivered with all the components of it, as we break down things, as we digest food, we're producing these byproducts, we have to get rid of them. We're also using that concept to maintain the proper balance between those things in water. And so this is another spot for that, because you've got the blood vessels exposed to some mechanism that would be now able to release that into the environment. So for fishes, a lot of it is at the gills, but for some of it, the nephrons and the kidneys function as well. Okay, so, and this is just kind of describes the process, and so, some of these things enter into the collecting tubules and the structures of the kidneys, and then water and some important ions then can get back, right? And so it really depends on the environment that the fish is in, how much of this is happening, right? So if you're living in one environment, you're trying to get rid of as much water as possible. If you're living in a different environment, you're trying to hold on to water. And so the specifics of this part kind of varies quite a bit among fishes. Okay, and then ultimately what's happening is that byproducts end up in the urinary bladder, and then that's going to be excreted into the environment. Okay, the bladder for a lot of terrestrial vertebrates, or most terrestrial vertebrates, I guess, is mostly just a storage mechanism. And so for us, when if you drink excessive amounts of water, your kidneys are going to get rid of it. It's going to go into your bladder. And then at that point, there's nothing you can do about it. right? So at that point, if all of a sudden you are cut off from your supply of water and you have to walk around in the desert, you're still gonna release all that water from your bladder whether you need it or not. We can't get it back once it gets into the bladder. For some fishes, that's not necessarily the case, right? So they're gonna use it to store, but they can also get it back into the bloodstream, depending on the conditions. And so the urinary bladder is important because it helps to remove salts from the urine in freshwater fishes, which is gonna be important because these animals are losing water all the time. But when they lose water all the time, they're also losing other stuff. And so you have to kind of get some of that stuff back because obviously you have to have, because if you're getting rid of a bunch of water and also a little bit of solutes every time, but huge volumes of it, eventually the solutes are gonna drop below what you need it to be. So they have to get some of that stuff back. It might also allow fishes to get water back. And because some of the fishes that are living in a marine environment, saltwater fishes, are in danger of being dehydrated even though they're living in water because of the surrounding environments. Okay, so the organisms, if we just look at the kind of organisms that are out there and what they do, if we're looking at the anathids, the hagfishes are mostly osmoconformers, which means their solutes to water ratio doesn't change a whole lot. But one of the reasons why that's true is that they typically live in places that doesn't change a whole lot. So they're relatively deep water animals. And so we talked a little bit about temperature. Water at 4 degrees Celsius is very dense, but also water with a lot of salt in it is very dense, right? So how many people have ever been to the ocean? Raise your hand. How many people have floated in the ocean? How many people have floated more easily in the ocean than you did in a freshwater lake? So salt water is more dense, and so it holds you up. If you have a lot of salt, it's going to the bottom. Right? So if you're a deep water marine animal living on the bottom like hagfishes, it's going to stay pretty much the same, right? So they're osmoconformers, but they're still relatively similar to the environment. So there's really not a lot of danger to them in terms of what happens to their osmotic concentration in the cells. Okay, for lampreys, lampreys are osmoregulators, and a lot of them move from marine to freshwater environments, so you're going to have to be able to do something. And so they're going to be similar to the teleos that I'm going to talk about in just a second. So we'll just kind of lump those in with the rest of the teleos, the bony fishes. For elasmobranchs, it's a little bit different. They are going to do a couple of things that do different things essentially for them. One of the things that these animals do is, instead of getting rid of nitrogen into the water column, because that requires a lot of water to be lost, these guys will convert the nitrogen into urea. And that does a couple of things. One of the things that does is it increases the concentration of solutes in the bloodstream. And that's important because it more closely matches the concentration of solutes in the environment. Right? So if you're an animal that you're swimming around in salt water, then the problem that you're going to have is water leaves the cells. But if you increase the salt content in the bloodstream, then now water is less and you lose less. And so that's one of the mechanisms that elasmobranchs use to maintain that. Of course, the problem with that is urea is still toxic, right? And so in addition to that, the elasmobranchs produce another molecule, TMAO, trimethylene amino oxide, and that particular molecule protects the cells against the toxic effects of urea. Okay, so what they're doing is they're taking ammonia, converting it into urea, which is less toxic, so they can have it in their bloodstream more than you can have nitrogen, ammonia, basically. but it's still toxic, right? So they have to also produce this stuff to protect against the toxic effects of urea. And so the overall result of that, because they're producing, or they're holding on to urea and they're producing this stuff, is that the concentration of ions in the bloodstream is a little bit higher than the surrounding water column. And of course, the bottom line from that is that they're losing less water than they would if they're swimming around in salt water, basically. And so the teleos, the saltwater teleos, don't have that mechanism, and so they have to do some other stuff. Okay. And of course, another thing that's kind of associated with that is that as these molecules build up, sometimes they get excess salt in the bloodstream, and so they've got some specific tissues that get rid of that specifically. Of course, that requires energy, and so it's not something that they can do passively. They have to actively excrete that from the bloodstream. Okay, let's see, what else? Okay, for the next group, for the freshwater teleos, these guys are hyperosmotic, and so basically there's different conditions that animals can live in, especially aquatic ones. All right, here's the fish and here's the environment. If you're a freshwater organism, you might have 10% solutes and then 90% H2O. If you're living in a freshwater environment, you're going to be 95% H2O and then 5% solutes. And so this animal will be hyperosmotic to this, right? So what's happening is, because the solute concentration is higher, that means the water concentration is lower. And so if this animal is swimming around in water like that, and passing a bunch of it over the gills with a huge amount of surface area, what's going to happen is this water is in a higher concentration in the environment, and that's going to seep into the plasma, right? So as these animals are swimming around in a environment where there's a lot more water on the outside, the cells start to absorb water, and of course that's a problem, right? Because why is that a problem? It disrupts what? Starts with an H. Homeostasis, right? So all the mechanism is adapted to this concentration of water to solutes. If water starts leaking in, then this goes up and that goes down, and then everything starts to shut down, right? So they're living in a particular environment that they're trying to get rid of all that excess water. And so essentially what's happening is they're gaining a bunch of water and losing solutes is another thing that can happen, right? Because the solutes are lower, the solutes are higher. Solutes generally don't move across the membrane as much. Some of them do, like ammonia does, right? And so that overall amount of solutes is going to be decreasing as they're exposed to different types of environments. And so some of the things that these guys do to counterbalance that is they excrete large volumes of dilute urine, right? So these guys, I don't know, and so what these guys display essentially are relatively numerous and large glomeruli. And the reason why that's important is because at that spot, a lot of the water is going into the Bowman's capsule. And so if you have a lot of these, then you're getting rid of a lot of water. Another thing that they have are ionoregulatory cells in the gills. And so what these guys, so ultimately what's going on is they're trying to get rid of all this excess water, and these things tend to leak out. And so what they're trying to do is replace those. As they're excreting lots and lots of water, now they're also decreasing their ions. And so they have some ionoregulatory cells in the gills that take up sodium and chloride ions from the water and actively pump it back into the fish. right? So all the things that we're talking about here are to regulate, or limit the loss of water or increase the amount of water that's getting out of the organism, and then replacing some of the ions that are lost directly to the environment, and also as a consequence of getting rid of so much water. Another, and of course this is another part of that, and so some of the ions can be recovered from the kidney filtering process. Mostly what these animals are doing is getting rid of excess water. Another thing that they don't do is they don't ingest water, right? And so in comparison to the marine organisms, these guys, even though water's going into the buccal cavity, it's all coming out of the opercular valve, right? So they're not ingesting water, because that would of course make the uptake of water worse. And so they're trying to limit that and excrete as much water as possible via the kidneys, and then just letting it come through the buccal cavity without going in. For marine teleosts, of course, you have the opposite situation. So these are animals that are, generally speaking, they have a lower amount of solutes than the surrounding environment, right? So in other words, they've got more water in the plasma than the environment does. And so physically, the same thing's happening. The water's coming in and being bathed across the gills, but what's happening with water and solutes is the opposite of what's happening with freshwater organisms. And so they're in a hypoosmotic environment, and so their problem is that they're losing water and absorbing excess ions from the environment. And so they have to do some of the other, some things that are different as compared to the freshwater fishes. And so one of the things that these guys do is they actually ingest water. So if you're in an environment, you're losing water, but you're in the environment that's water, one way to counterbalance that is to drink it. Of course, the problem is what?
They're full of salt.
Yeah, you're also drinking salt, right? So you're replacing water, but you're also increasing the uptake of salts by doing that. And so they're gonna have some other mechanisms for that as well. So they also have ionoregulatory cells in the gills, but what they're doing is getting rid of that because they've just ingested more than they need because they're drinking the salt water to try to replace the water. Water is also recovered from the kidney and also the bladder, right? So this is a mechanism that these guys have that other vertebrates don't have. Once water gets into the bladder, they can now resorb it back into the bloodstream because their problem is losing too much water. They also have a different arrangement of their kidneys, so much fewer, or many fewer, and very small glomeruli. And the only reason why that's true is because that's where water gets lost, right? So if you have a large glomerulus with a lot of capillaries, then if you expose it to a Bowman's capsule, it will lose water. And so there's no mechanism for stopping that process, but you can reduce the number of them and you can reduce the size of them. And so overall, the amount of water that's lost at the kidneys is going to be a lot less. And of course, that would be important to the marine animal because they're constantly losing water to the environment. Okay, let's see. I think that's it. Okay, does anybody have any questions about that?
Where exactly is the line between exam 2 and exam 3?
Exam 2 is going to cover what I just finished. No sensory systems on exam 2. Does that make sense? Okay, yeah, so normally I would.
This is all.
Did
We're
have-
fixing to start with these.
So all of that will be on exam 2.
Yes. Yes, well, not all of it, but some, yeah, gotcha, some questions from any of them. Yes, your exam will be 7 hours long. Okay, so that's it. And your test is sometime
Tuesday?
Tuesday.
Where's it?
Thursday.
Thursday. Is it
Thursday.
Let's go for Thursday.
Is it Thursday?
Thursday.
We can make it Thursday.
You guys are,
Thursday.
y'all are messing with me.
I say we do it Thursday.
It's scheduled for Tuesday.
But.
Can we do Thursday?
I'm open to having it on Thursday.
Thursday.
Thursday it is.
Nah, it's Tuesday. Let's see, I don't see any reason why we wouldn't have it on Thursday. You guys okay, everybody okay with having it on Thursday? Thursday.
Yeah.
Anybody opposed?
No.
Usually not. Huh? We're not done yet, I still got 12 minutes.
You got 2 minutes.
Yeah, 3 minutes.
Oh, never mind.